Hi, I'm Mike Travers from the SRI Bioinformatics Research Group. This webinar is going to teach you about web groups, a relatively recent new feature on the Pathway Tools websites. Web groups let you create and store sets of genes, metabolites, and other biopsych objects. Groups can be shared with your colleagues, and you can perform various computational analyses and transformations on groups, such as enrichment analysis. Groups can be generated from within biopsych, for example, from search results, or you can create them by uploading your data from a spreadsheet. You can start using groups by choosing the Groups command from the Tools menu. We'll do that. And that takes us to the group directory, which lists all the groups that are accessible to you. Now in this case, we aren't logged in yet. So all the groups I'm seeing are public ones that people have shared. To see my own groups that I've created in the past, I need to log into my account. Now you can log into the account up here, clicking on the login link. Now I have an account already. If you don't have an account, you can click on this Create One link and it will take you through the process of creating one. But I will log in. And now we see the directory has some more tabs, including this My Groups tab, which lists all the groups that I have created and are private to me. For our first task, we'll create a group of genes from experimental data, explore some of their properties, and take a look at several methods for finding related pathways. There are three different ways to do this using groups. First, by using transformations, second, by visualizing them, and third, by using enrichment analysis. We'll try all three over the course of this example. And we'll do it by uploading data from a text file that we've created, and we'll take a look at the text file first. So this is just simply a list of E. coli genes, one per line. And to create a new group, we go up to the Groups menu here, which lists all of the Groups commands, and we'll say New Group from Uploaded File. That puts up a dialog which lets us specify the file, which is this one here, and a few parameters, and we'll just click Upload and create the group. And there it is. So we've created this group. Now, a few words about group structure. A group is very much like a spreadsheet. It has rows and columns. In this case, we just have one column, and we have 120 rows, which correspond to the genes from our file. And each cell of that column contains a gene object that points back into the knowledge base. So we can change some of the names of our group by clicking on them. So let's give the group a better title. RP differentiated genes. We can also change the name of columns in the same way by clicking on them. So typically, a group might have several columns, but the first column will contain the chief objects of interest, in this case genes. But we can create other columns that will unveil relationships or properties that relate to those objects. So now we'll add some columns from the Property menu here. So this is a selector. This shows all the properties that apply to this set of genes. So we'll pick synonyms. So that creates a new column, which for each row will show any known synonyms for that gene. In some cases, there's more than one, like in this first row. In some cases, there's none. We can also add, for instance, the accession number. And once we have these columns created, we can do things with them, such as sort. So we'll sort by the accession number. That gives us these genes in that order. So transformations, in a similar fashion, create a column that shows a set of related objects. For instance, we can use a transformation to see the protein products of our genes. So that's accessed by this other drop-down menu here, and we can see that these are transformations that apply to genes. We'll pick the products of gene. That creates a new column that contains the protein objects that those genes produce. Now, one thing to note is that all of the properties and transforms we've done so far are based on the first column, but they don't have to be. You can select other columns, and you can do that by clicking in the header. You'll notice that that highlights now. So that's now our selected column. Now the transforms and properties apply to that column. So we can, for instance, see properties of the proteins, including the molecular weight. So let's add a column for that. And now we have molecular weight of the protein product of the gene, and we can do operations on that, such as sort. We'll sort from highest to lowest. So now we'll look at the first way to find related pathways using transformations. First, we'll reselect the gene column, and then we'll pick the transformation to find pathways of those genes, which is there. And that's off the screen, so we'll delete some of these other columns. Now we see for each gene, it lists the pathways of those genes when they're known. I should point out here that the web group system contains a large collection of transformations. We encourage you to explore them all. Please keep in mind that the list of transforms available, it shows up in the menu here, depends on what type of object you've got selected. So for the gene column, these transforms are available. If we click a different column of different types, such as pathways, a different set will be available. So please try them all. Now, the second way of visualizing pathways that are related to a set of genes is by visualizing the genes on the cellular overview. So to do that, we'll go back and reselect the gene column, go up here to the Groups menu, say Paint Data on Cellular Overview. And that brings up the cellular overview with the genes in our group highlighted. So now we can visually explore which areas of the cellular overview are highlighted. If we zoom in far enough, it will start to show you the names of pathways. So for instance, here's a pathway that contains a few highlighted genes. The third way to find related pathways from a group of genes is through enrichment analysis. So enrichment analysis is a statistical technique for finding explanations for sets of genes or other objects, often, but not necessarily from experimental data. In this case, explanation means objects or concepts that are linked with the original group at statistically significant levels. Web Groups offers a variety of enrichment analysis tools that are available from the enrichment menu. For instance, given a set of genes, we can enrich that set for pathways, that is, find pathways that contain more genes from our set than we would expect to occur by chance. So let's do that enrichment. We'll select that command. Pops up a menu of some options that control the details of the statistical computation. And we'll just take the defaults. Now this is going to create a new group of pathways. There it is. So some things to note about this group. So the first column contains both individual pathways and pathway classes, which are distinguished by the use of uppercase initial letters. Second column is the p-value, meaning the statistical significance that it's found, and the group is ordered by p-value. So the pathways at the top are the ones that are most overrepresented. And it also shows you the genes that matched from the original set. So I'm going to delete that match column right now just so we can see the pathways a little bit better. So there they are. So we see that these are all the pathways that enrichment analysis has produced, and we can see a lot of them relate to biosynthesis of amino acids. So that's something we've learned. Okay. For our next example, we're going to do something a little bit different. In this case, we're going to create a group entirely within biocide. So we're going to determine the set of genes regulated by a particular E. coli protein, and we'll learn something about them. So let's start by finding the protein that we're interested in, and we can do that using the quick search box. So here's some objects. This is the protein we're interested in. ARC-A phosphorylated DNA binding transcriptional dual regulator. So we'll click on that. Go to its object page. Now this is a standard object page, but now there's a new feature on this and all the other object pages, an Add to Group button. So if you put your mouse over there, that expands a bit, and it sees some options, gives us some options. We can take that object and add it to an existing group or make a new group based on it, and we'll choose the new group. So add that. Now it's created a new group, and it's left us a link to that, which we can click on. So now we go to our group. So here's a group. It's got one row, one column. Not that interesting in itself, but we can use transformations to build new groups based on that. So we will choose the transform genes regulated by protein, RNA, or compound. That goes off. So now that creates a new column with all the genes that are regulated by our ARC-A protein. There's quite a few of them. Now in this form, they're all jammed into one cell of a group, and they're not that useful in that form. But we can take that set and make a new group out of them, and that's what this green button on the top of each column does. What that does is take all objects in that column and makes a new group out of them. So we click on there, that will make a new group of genes where each gene is in its own row. It shows us the matching object, which is not too interesting because we only started with one object, so that's going to be the same for all genes. So we'll get rid of that. Maybe we'll change the name of this to something a little easier to deal with. Genes regulated by ARC-A. Now from that, let's say we want to see what pathways those genes appear in. And we saw enrichment analysis, one way to do that, but we can also use a transformation. And this will show pathways that have been curated to include that gene. And then we can do the same thing we did before. We can make a new group of those pathways. There we are. So now this is a group of all those pathways, and here the match column is a little bit more interesting. It shows us the gene in the group of genes where that pathway matched. And in many cases, there's more than one gene that matched. We'll show one further way to create a group from the results of a search. So first, let's do a search for short E. coli proteins. Go to the search page, and we'll filter by sequence length, and we will say the amino acid sequence is less than 100. Do the search, and there's the results. And notice there's a button on this search results and all search results that says turn this into a group. Click on that. Now those search results have been put into a group where we can manipulate them. So now that we have these, we can do our usual things with them. Let's start doing some enrichment analysis on this. So we'll go to the enrichments menu. This time we will enrich for Go terms. So we'll choose genes enriched for Go molecular function. We have to pick the default parameters, same as last time. And there we go. So this is a group we haven't seen before, where the elements in the first column are gene ontology terms, but that sort of illustrates that a group can contain any kind of biopsych object. It also shows the matching p-values and the genes from our gene set that match that. So let's delete the match column just so we can see our results a little bit better. And so these are the gene ontology terms that are overrepresented in our group of short proteins. And we can see it's kind of interesting. These seem to be a lot of RNA and DNA binding functions in there. So this tells us something a little bit about our genes. Now, one other thing we can do with this, once we have this data, we can export it to a spreadsheet through the Groups menu, export to spreadsheet file. Click on there, and that downloads a file, which you can then click on, and there it is in Excel, and re-import it or send it off to another application. So that's probably enough about groups for this webinar. WebGroups has many more features, including the ability to filter groups, to share results, to perform set operations on groups, and manipulate genomic sequence data we will cover that in a subsequent webinar. Thank you for watching.